great speaker, Professor Olivia Eberling from Baylor College of Medicine. Before introducing her, I would like to go to our center directors to see that whether they have any update that they wish to share with us, starting from Professor Janet Roberta from University of colleague at Baylor College of Medicine that I have the pleasure to introduce her is Professor Olivia Eberling. Livia actually, her training background is in chemistry and biochemistry. She received her PhD from Purdue and bachelor from Campinas University. And she became soon interested in area of biomedical. She got her postdoc at the Stanford University. Then she started her faculty position at the University of Texas in Austin, and currently is an associate professor at Baylor College of Medicine. Livia's research project is focused around development and application of mass spectroscopy in health-related research, with a particular focus on disease detection and diagnosis to improve patient care and clinical outcomes. She received many awards, and it's amazing. Probably good to know that she's listed in the Forbes 30 under 30 in the health category. And also she received more Inventor Fellowship as well as MacArthur Fellowship in 2018. She's leading now our Surgical Innovation Unit at the Department of Surgery at Baylor College of Medicine, and already make a great actually impact at our department. So it's a great pleasure to introduce Livia Eberling. Livia, the platform is yours.
Thank you so much. I really appreciate the opportunity and the enthusiasm. I'm really happy to share with you today not only about our program at Baylor College of Medicine Department of Surgery, but a little bit too of my background in innovation and how was it that a chemist is now doing surgical research and translating technologies to the clinic. So I really hope to be able to convey that to you today. Can you see the presentation mode, or I think I got it. Okay. So as Dr. Najafi said, I just recently moved from the University of Texas at Austin, where I was an assistant professor in the Department of Chemistry, to the Department of Surgery at Baylor College of Medicine. And most of my colleagues in chemistry were a little bit surprised with that decision to make that move. But for me, it really felt very natural and what I've been wanting to do for a really long time. As Dr. Najafi said in my introduction, even though I'm trained as a chemist and I'm actually trained as an analytical chemist, I've been doing medical research for a really long time and had the opportunity to work side by side with surgeons and pathologists, and through those interactions identify needs for which chemical analysis could really advance how medicine is performed and how we care for patients, especially for our patients with cancer. I started doing medical research at the Brigham and Women's Hospital. That was primarily focused on neurosurgeries, and that was during grad school. And that was when I really was primarily going to operating rooms to collect samples, and very nosy, very curious about everything. I would just stand there waiting for samples, either in the OR or in the pathology room. And that's when I started to observe that some of the techniques that were being used, for example, frozen section analysis, have been used in the clinic for almost over actually 100 years. And that's when I thought, there's just so much innovation that's being done on basic laboratories, but there's really kind of like missing a path or a pathway to actually translate and adapt these technologies in a way that can actually be used by medical professionals, and in the case of my research, by surgeons. So I really hope that is conveyed today, and I'll tell you a little bit more about how my background has really enabled me now to be in the Department of Surgery. So backing up to 2004, I was an undergraduate student in Brazil. I'm from Brazil. And at that time, I was very fortunate to start undergraduate research with mass spectrometry. And I know how many of you have heard about mass spectrometry, and I'll tell you a little bit more about how the technology works. But at that time, which is now almost 20 years ago, I was using this very complicated instrument that you can see here in this picture. This is a real picture of the system that I used to use. It's a in vacuum kind of physical chemistry kind of experiment and equipment. I sat right in front of this panel here with several knobs. We optimized the voltages in every single piece of ion optics, making sure that we got really good sensitivity for specific ions. And at that time, I remember very clearly that I was working with this gas phase ion molecule reaction and trying to understand what are the intermediates in this chemical reaction. And I was fascinated by the ability that mass spectrometers had in measuring molecules. I thought that was really remarkable that we could put a chemical compound in the system and get a spectra or a graph that showed you exactly what those molecules are. But I was also very kind of bored and like not really excited about it. Was like a very unique gas phase reaction, not even liquid phase, that wasn't really applicable to anything that I could see an immediate impact. But that's what we had. Now that we're in 2022, the technology has advanced so tremendously that mass spectrometers have gone from these very bulky, difficult to use systems that you kind of have to babysit and take care and change a lot of stuff daily, to systems that are truly clinically appealing and that could be translated to the clinical setting for use by medical professionals. So for example, the systems that we use today, this Orbitrap Exploris, even though it's still big and a bit bulky, it fits on a table. It can be rolled into an OR. It can be rolled into a pathology room. It will work on batteries. So it has a lot of the features that we need to use this technology in the clinic. We also have mass spectrometers that are the size of a shoebox. So they're very small. There's some mass spectrometers that can be put on backpacks. They do not have the same level of performance for molecular analysis. They're restricted to a few molecules on lower resolution, but still, when you're looking at analysis of specific molecules and compounds for field use, these are very appealing platforms. And really, these developments in the field have enabled now us to develop devices and technologies integrated to mass spectrometers that we can actually put in the hands of surgeons and clinical professionals. So the vision in my group is that we want to develop and translate mass spectrometry technology to improve patient care across kind of all stages of care for patients. So in research laboratory, when we're doing basic research, discovering biomarkers, mass spectrometry plays a huge role. That's actually where you will find mass spectrometers in research laboratories. People are using it for proteomics, metabolomics, lipidomics, really trying to understand disease development and so forth. In the clinical lab, so clinical microbiology labs or clinical laboratories, you do find mass spectrometers as well. Those are primarily used for targeted assays. You look at metabolism or blood analysis as well. And they are part of FDA-approved clinical assays, but somewhat limited. In the pathology room, not as much. All of the mass spectrometers that I know that are in pathology rooms are part of research studies. So there's still opportunity there to incorporate mass spectrometers in pathology room to analyze biopsies, tissues, and so forth from patients. And I'll tell you today about our work that I would say is the most disruptive in the operating room, where we're putting a mass spectrometer in a place where you don't really see mass spectrometers at all, right? You see PET scanners, CT scanners, ultrasound machines, anesthesia machines, robots in the operating room, but not really mass specs. And really what we're trying to bring mass spectrometry and molecular analysis to the hands of the surgeon. And my whole vision with mass spectrometry technology is that we would be able to do direct analysis of tissues that is rapid and unmodified samples without having to do extraction, separation, and a lot of sample workflow that is normally used in mass spectrometry. Combine that direct analysis with high performance and smaller movable mass spectrometers that can be used in the clinic, and also use advanced data analysis systems so that we're using AI and machine learning to interpret this very complex molecular information that we acquire from biological samples and provide the clinicians with something that's clinically actionable. So we're not only acquiring this very chemically molecular information, but we're converting that information into something that's clinically actionable. So this really is a summary of primarily everything that we do in my lab. Just wanted to do is mostly focused on metabolic information. And when we think about clinical diagnostics, a lot of the times we think about DNA and RNA or protein analysis. And I just want to stress out that the ability to measure metabolism directly from clinical samples is really exciting, really powerful, and we can correlate metabolism to diagnosis, prognosis. We can look at subtyping of diseases and tissues. We can also look into response to therapy. We have projects where we're using metabolism to inform if a patient will or not respond to immunotherapy, chemotherapy. And then we can always correlate these patterns of molecules of metabolites to gene expression, to protein expression, so that we can further expand the analysis that we're doing. So why so good for analysis of metabolites? Mass spectrometry is not spectroscopy. It's not based on light. We're actually measuring mass of molecules. And these molecules are not only restricted to one specific compound, but with mass spectrometry, we can do untargeted analysis of hundreds, if not thousands of molecules in a single reading. And this really provides unparalleled sensitivity, but also chemical specificity to our analysis. This is primarily due, again, to the fact that what we measure is the mass of the, so here you can see in the x-axis, this is mass to charge. Most of metabolites will have one charge, maybe two. But if you have one charge, then it's the mass divided by one. So we're primarily measuring mass. And because we cover like quite a range of masses here, we often measure from 100 to up to 2,000. Then you can imagine how many molecules will fall within that mass range, right, from their molecular weight. And with the resolution that we achieve with the mass spectrometers today, that's way beyond that one example I showed you in my undergrad. Now we can detect molecules even that have the exact same nominal mass, but have different accurate masses. So two different lipids that may weigh 816 Daltons, they will be separated by mass spectrometry if they have different accurate masses. So up to like the third digit after the point. So that really provides us an incredible possibility to directly analyze these samples and detect hundreds of molecules that are naturally occurring in these. So just want to show you an example of the molecules that we focus in our analysis. We're thinking about primary, secondary metabolites, things that are involved in the TCA cycle, very much related to metabolism, very much related to respiration. We look at amino acids, purines, and polyamines. We look at fatty acids, sterolipids, derivative, lysolipids, and a lot of glycerophospholipids that are crucial to the integrity of our cells. They're in the cellular membranes, but they're all also signaling molecules as well. So these molecules, or a panel of these molecules, are very informative of the disease type that you're analyzing, of the tissue type, and can be really used for diagnosis and prognosis and typing it. Let me jump into the technology that we developed in my lab. We started this work at UT Austin. So as soon as I started my independent career in 2016, this was our very first project that we tackled. And here what I wanted to do was, I knew that mass spectrometry provided, right, this incredible ability to record molecular information with high specificity and high sensitivity. What I felt was really missing was that interface between a user that had no clue about how mass spectrometers work and the actual mass spectrometer. So how could we fill that gap? So my idea was, well, we need to develop something that is perceived as being simple, that can be used by anyone that does not have training in analytical chemistry. We need to make it into something that looks like a device that you could just grab, use, and you could dispose, or you could reuse it, and that wouldn't require optimization. It wouldn't require high voltages, which is normal in mass spectrometry. So this was really my motivation to developing what we call now the Mass Spec Pen device. You see this here, this was our very first prototype. We fully 3D printed this in my lab. We really paid attention to working closely at this time. At this time, I was collaborating with surgeons at Baylor College of Medicine and at MD Anderson, and really getting their insights into what are some of the features into a device or a technology that would make this something that you'd want to use. And I like to stress that because I'm sure all of you here today are inventors, and you think about technology development from a user perspective. And I think basic scientists, not as much. We like to make things often very complicated. And this was really the opposite of what I was going for here. I wanted to make this the least complicated possible so that we could enable use within the medical environment. So the mass spec pen, I'll show you how it works. It performs a very gentle extraction of molecules using a solvent droplet. It's non-destructive to the tissue, which I think has been truly transformative in being able to translate this technology. We designed it with the intention of being used in vivo during surgery, but we have really analyzed many tissues, ex vivo, biopsies, and a variety of other samples, like infected samples. I'll show you a little bit about that. And I really want to make it into a plug and play, user-friendly system that the user wouldn't have to think, again, about optimization or anything that was technically challenging. So I envision it to work, which is how it works today. So imagine the surgeon is holding this device, the mass spec pen device. It is connected to the mass spectrometer. We've recently actually filed a patent disclosure on the detached version for field use. Now, I'll mention that later. But the whole process is triggered with a click of a foot pedal. So that was also insight that we got from the surgeons, that ideally they want to just click on a foot pedal and have the whole process triggered. So we enable that using microcontrolling units. So we use a Arduino microcontroller, and now we're using a Raspberry Pi as well. This high floating control, perfect. So it's very intuitive to use, and everything is automated. So there's no need for any triggering on a computer or anything like that. So once the surgeon touches this pen or this device onto a tissue, we designed this tip, which is made of polydimethyl so PDMS. It's a biocompatible polymer. It's used in a variety of medical implants, super safe for touching the patient. We sterilize everything before, of course. So in this tip, we have 3 channels, and then we use the first channel to deliver a controlled volume of solvent into this reservoir. The reservoir is really important because it keeps that solvent within this environment, this volume, 3-dimensional structure, and it allows the solvent to interact with the surface of the tissue in a way that we extract molecules. We know solvents extract molecules. This is a basic chemical principle. I tend to say that my coffee is a chemical experiment because I use water to extract molecules from a solid sample, which is that coffee powder. In the same way here, we use water to extract molecules from a solid sample that is a tissue. And a lot of times people get really puzzled of how this happens, but it's really something that we do routinely in our laboratories, of course, but also part of our lives. So volume of solvent in the OR, we primarily use sterile water because of biocompatibility, but we've used a range of solvent systems. I'm a chemist. I love playing with solvents. In the lab, we use a variety of different organic solvents to change the extraction capabilities, but in the OR, we use water. And it's just really easy to tell the surgeons that it's water. So once these molecules are extracted into the droplet, then we use a vacuum suction to transfer this droplet through the tubing system to the mass spectrometer. Right at the interface of the mass spectrometer, we have a heated source, which is literally using just a heating system to vaporize these droplets. So everything that's in gas phase, including these molecules that were in the droplets. And then these molecules that are charged go into the mass spectrometer. And then the mass spectrometer does its job. It records these incredibly rich molecular profiles, including hundreds of metabolites and lipid species that are then informative of the tissue analyzed. Unplug these pens and we can throw them away. They're all plastic materials, or you can sterilize it and reuse it. We do not do that because of the IRB. Currently, the IRB protocol does not allow us to do that for patient safety. But in the lab, we do that. In our lab, when we're using just excised tissues, we autoclave and use it again, or we just wash it and use it again. Okay, so a closer look here of how it looks. I already explained it to you. Since we are using 3D printing, we've been pretty creative with the size of our device. We've made minimally invasive probes, and I'll show you the work we did with Intuitive for the Da Vinci Xi robot, but we also have gone up to like 10 millimeter probes. This is for some of our breast cancer lumpectomy studies where the surgeons wanted to look at a larger area. So we can adjust this in our CAD draws and just print them in a different diameter. We do place the pen on the tissue for about a 3 seconds period, sometimes 5 seconds, but the whole process is completed in about 10 seconds, 15 seconds. It depends on the length of the tubing. And these are the beautiful profiles that we detect. And this may look to you like a complex graph, but it's not. This is actually molecules being measured from a tissue. These are the mass spectra. And X, again, is the mass to charge ratio. Remember, I said most of the molecules have one charge, so this ends up being the mass of the molecules. And then Y, it's their abundance. And what we have observed through tens and tens of studies, not only from my group, but from our field in mass spectrometry, is that these profiles are very robust, very reproducible, and there are changes that not only related to cancer in normal tissue, but we can subtype these cancers. We can say this a low-grade tumor. We can even correlate with genetic mutations. We're able to identify, for example, IDH1, IDH2 mutant tumors and brain tumors. This has huge implications for prognosis. So we can use this information and dig deeper into the molecular information to really enable stratification of disease. And you can see here, I identified a few of these peaks. These little peaks here are not noise, these are molecules. And as we dig in this mass spectra, you see more and more molecules, but you may recognize here some glutamate, ascorbic acid, lactate, glutathione, fatty acids, and then many lipids as well. Okay, so one of my favorite features about the method is that the analysis is fully non-destructive from a histologic perspective. So this is a tissue before, during, and after analysis. And even though we are extracting these molecules, this is very gentle. So just imagine putting a solvent droplet on the tissue and then aspirating it. There's no like physical damage that is macroscopic, but neither microscopic. We've worked closely with pathologists and they've looked at sections of the tissue right where we analyze, and there's no damage. So this really enables us to incorporate this technology and this molecular information into current clinical workflows, to where we don't need to tell the pathology staff, we need an extra piece of tissue, or we're going to completely consume this tissue. We can do the analysis, and then the samples, they keep going through the sample workflow for routine clinical analysis. So, and first reported in 2017. Since then, we've really expanded to a variety of different cancer types. Our latest publication was in Whipple procedures for pancreatic cancer last year. We're working on our publication for breast cancer and thyroid cancer as well. And the features are really kept the same throughout all of these tissue types and disease types. The analysis is non-destructive, rapid, and we've kept an accuracy of about 96% through all of these analyses. So this table will really break down by specificity, sensitivity, accuracy, and the area under the curve for all of the classifiers that we developed. So let me just touch a little bit on the classification systems. I'm not going to go deep into the algorithms that we use. We do use logistic regression to go from these huge data sets and shrink down to the predictive features that are most indicative of the disease state. We train our classifiers. We really love using the Lasso algorithm that was developed by Rob Tibshirani. He was one of my mentors at Stanford. What we really like about the Lasso is that it not only provides classifiers, right, like you think with machine learning, but it also has a penalty function that shrinks your classifier to the most predictive features or ions or molecules for certain classes. And that's important for us because, as I said, we can start from data that has thousands of molecules, and we don't want to just blindly use all of these thousands of molecules for prediction. You want to be able to find within these thousands of molecules, what are the molecules that are truly valuable in identifying a certain class, right, of disease or tissue. And this is really what the Lasso does. As we say, it's like a shrinkage operator because it shrinks the data set to these predictive. We've developed a graphical user interface where a user could say, well, I'm analyzing ovarian tissue. This is an ovarian surgery. And then you analyze that tissue. We acquire the mass spectral data. As I said, it's really quick. This is like 10 seconds or less. And then our classifiers are already running in this algorithm, in this platform to provide a diagnosis from those markers. And we've developed these with training sets. We did cross-validation and we validated with an independent data set. And now we've been testing it independently in the operating room. But there's always room to improve algorithms. I'm sure many of you work with machine learning and AI. There's always the possibility of overfitting to a training set of data. So we're constantly refining, testing, modifying, and really making sure that our predictors are robust. They can perform well in independent data sets for implementation. So Grey's Anatomy is just a really cool video. It wasn't perfect, but they did a pretty good job, and I thought it would be fun for this audience for you to see this.
It really on the testing stage. We're testing on this guy. He's got a wife and 2 kids. Dr. Gray, if you could just tell Dr. Wilson.
Be quiet. Now you get to go tell his family that not only did you saved his quality of life. I'm gonna go check on our stoners.
An example, and actually they showcase our intended use, which was help with surgical margin evaluation, which is critical in the removal of several solid tumors. So we actually started a real life Grey's Anatomies in the end of 2018. We had a really good run for like a year until like the end of 2019, going into 2020. And then of course, COVID happens. We had to stop all of our clinical studies for almost a year. So that was really something that delayed progress in our clinical studies. But since then, now we've really picked up and restarted a lot of these projects. So we were able to do this primarily through my incredible collaborator, Dr. James Stollerberg, who's been a surgeon at Baylor College of Medicine for many, many years. And he's really helped us through all of the logistics of installing instrumentation and getting this study into place for testing with patients. Best news of all is we had no reported adverse effects for any of the patients enrolled. We have about 180 patients now in our studies altogether. And this was really something that was very exciting for us. So just showing you some images. This is Dr. Stollerberg using it in a thyroidectomy procedure. We do analysis in vivo. We also analyze the tissue ex vivo once it's excised, and really like comparing the data, you know, how does it perform in vivo? How does it perform ex vivo? What do we need to improve? How do we revise the design to improve it for analysis? So yeah, I just wanted to point out, I didn't want to go deep into the science, but I wanted to just point out this study, if you're interested, that was our first reported diagnostic study. This is for pancreatic cancer and Whipple procedures. Dr. Van Buren and Dr. Fisher at BCM used the system. Here, reminding you, this is the surgeons are doing the analysis. We are in the operating room helping for the cases that the patients consented, but it's in the hands of the surgeons. They are doing the analysis. They're doing the chemistry. I tell them this, you're doing the chemical experiment, even though you're not thinking about it that way. So that's really fun. So we really broke down the performance here by the type of margins that we evaluated in vivo, ex vivo. Our overall accuracy was 93.8%. And this is using pathology as gold standard. What we found very interesting is that as we acquired this intraoperative data, once we incorporated some of that into our classifiers, our machine learning algorithms, we actually improved our performance. We're not sure exactly why here, if it's really an effect of the OR data or just more data. So we're still investigating this, but this was really exciting to observe. We are just about ready to publish our breast cancer study. And here are some of the incredible surgeons that have been helping us with that. Dr. Carter, Dr. Thompson, Dr. Boniface, and then a pathology for Dr. Naji. And again, here we do analysis in vivo in the surgical bed, excised, and then on the excised tissue as well. We do have one of our systems at MD Anderson. This is the PI of the study here, Dr. Nicole Fleming, for ovarian cancer surgeries. Here is not really a surgical margin question. It's the ability to identify metastatic ovarian cancer within the patient's body and really helping the full debulking surgery to be more effective. So for this study, you can actually see our system. This is the mass spectrometer in our table. This is the battery here. We're now putting this battery inside under the table here, so it's all within one unit. and it will run on batteries for about 45 minutes. It does use a lot of power, this instrument, it's under vacuum, but it gives us sufficient time to roll it in and roll it out so that it's not constantly installed in the operating room. And here's Dr. Fleming analyze an excised piece of ovarian, peritoneum tissue. And lastly, I just wanted to mention that because I love prototyping, I love 3D printing. We do a lot of that in my lab. We had a collaboration with Intuitive Surgical and Thermo Fisher Scientific, and they sponsored this study. And we designed a drop-in probe of the mass spec pen that could be adaptable and integrated to the Da Vinci Xi system. So here you see that's a surgical robot, and I think I have a video. This is our redesigned mass spec pen being grasped here by the robot and placed on, this is a pig, placed on the stomach of the pig for molecular analysis. I just want to highlight there are a lot of other projects that we have been pursuing. I'm a chemist. I love chemical analysis beyond medicine, even though my passion is in medical projects. But I would like to highlight our most recent project in infectious diseases. So we have been working with Dr. Trotner on the diabetic foot infection and being able to directly analyze the infected tissue and identify molecules that are indicative of the bacteria. So we can identify what is this infection and expedite clinical decision-making in terms of the therapeutic options for the patients. And these are some of the other projects that we're involved in: clinical toxicology, where we're detecting drugs in skin and different patients. So some exciting projects there. I want to thank all of my collaborators at Baylor College of Medicine and at MD Anderson. Thank our research funding. There's a lot of support from the NIH and CPRIT and some foundations, but also from companies that have believed in our work and provided funding for specific projects. Just a little bit more now on our program. We founded a company around this technology in 2019. So this is me and James, my collaborator, James Sullerberg. And this was through this company formation process, which was something completely new for me at that time. I really got more involved into how do we patent things? How do we commercialize things? I work closely with UT's Office of Technology Transfer, and we also work closely with Baylor College of Medicine Ventures team. So it provided me a lot of background information. How do we go from a lab prototype to something that can be spun out into a company? How do we raise money? What's the kind of expertise and knowledge that we need to know? So based on that, Dr. Rosengart recruited me to come to Baylor College of Medicine, and one of my roles is to lead our Instinct program, which is really our incubator at the Department of Surgery. And our goal is to support our members in developing, testing, and commercializing their creative ideas into what we hope will become groundbreaking medical technologies, which is our hope for the mass spec pen too. Even though it was a really hard process and that takes many years. We really know that our ideas can be further developed with the appropriate support. So our mission is to also instill innovation as a core value into our department members, and to foster that kind of mentality in the development of new medical technologies. We know that a lot of people have ideas, but many times they just don't know what to do with them, or they have a prototype that they don't know, okay, how do I even submit a patent application, or how do we form a company, or do we license it to a larger company? So these are some of the questions that we're hoping to help our members with. And we do offer several levels of support. We support intellectually. We also provide financial support to early stage ideas. We help with designing and prototyping, and then administrative and networking support as well. We're working very closely with BCM Ventures in our institution so that we overlap with them well, and we transfer this idea from a prototype and something that's been testing into the hands of the college so that they can further develop it into through the licensing and startup formation path. So as I said, we do support our members in many different ways, and I just wanted to highlight some of our upcoming events. So one of the events that we are relaunching now is our think tanks. The first one from the relaunch is going to be this next Tuesday at 4 p.m. This was all started and initiated by Billy Cohn, who's an incredible surgeon and inventor. He's now at J&J, and he has an appointment in surgery as well. And these think tanks are really opportunities for people to come with new ideas or problems they have identified and just brainstorm and get insights from the folks in our department. And we will have a true Shark Tank event. That's going to be in October 19. First, we thought to make it like a mock event that you just kind of came with an idea that maybe you thought about, and then there was going to be a panel of investors that were not real. But now we're actually making this into a real deal. There's going to be real money, real cash for companies or for groups or teams that are developing something exciting surgery. So really looking forward to that. And just want to highlight our board members, including Dr. Najafi. Thank you again, Lee-Jung, for this opportunity to speak today. But these are our board members. We have a new resident that has joined our board to kind of be that connection to the residents in our program. And yeah, so we're just really excited to restart Instinct and help bring new medical technologies to reality. So if you want to check out our website, we're making these interviews with some of our successful inventors in the Department of Surgery. These are going to be short videos, just fun videos to, again, kind of instill that innovation mentality into folks in our department. But if you want to check out our website, and our events will be there as well. So I think this concludes my slides. So I just want to thank you again for the opportunity. And yeah, so excited to take any questions that you may have right now.
Wow, amazing, Livia. That was outstanding. So I remember that a decade ago when I started in College of Medicine, if you're saying that you have an idea that you want to commercialize it, that was taboo. That's conflict of interest. No, you cannot do that. But now saying that the university, they're trying to encourage them and even support them to commercialize the idea, having their startup. And this is the way that can make an impact. And I'm so happy also to see that engineers, other disciplines, that are coming directly to College of Medicine to see the unmet need and trying to actually address those unmet needs. Because I do agree, engineers, I'm engineer, we often tend to over-complex the solution, but sometimes the solution, what we are looking for is much easier. Just we need to build this collaboration and understand the problem. I know that several people in the chat that came up actually with many ideas. I said that is a genius idea. I mean, we talk about bringing care to underserved location as well. I remember that, for instance, for diabetic foods, one question that we have is that how much we should debride wounds, because we don't know that this tissue is an infected tissue, dead tissue, we should remove it or not. Losing too much tissue is really bad, but if leaving the tissue that is damaged, also it doesn't help the wound healing. Sometimes even detection of osteomyelitis is very basic, and definitely your technology can help not only in the OR, also maybe in the different location, any places that it can help the data. So I'm going right away to the questions, and Mike asked that can chromatography be combined with mass spectroscopy for more effective detection? Mike, please open up your mic if you would like to elaborate on your question.
I can answer that though.
Sure, go ahead.
So Michael, thank you for that question. So traditionally, mass spec is often combined with chromatography. So it's either a liquid chromatography mass spectrometry or gas chromatography mass spectrometry. And to be completely honest, I wanted to stay as far away from chromatography as possible because it is a time-consuming assay. So even though you do get quantitative analysis, and with gas chromatography or liquid chromatography, we're talking about thousands of ions, thousands of molecules that have been quantified. So the performance is definitely superior than doing mass spec alone. But what we have found is that the data that we currently get with just mass spectrometry is already sufficiently informative for the problems that we are pursuing. So we don't need chromatography because it's so time consuming and can be very finicky, I would say, any type of chromatographic column or anything. So we would rather just do direct mass spec analysis. And that really expedite things and provides us sufficient molecular information for diagnosis. But yeah, if you do chromatography, you get a lot more coverage, you get quantitative analysis, and it's really powerful for discovery.
That's amazing. So I know Mike is one of our industrial members. Maybe you can team up and help to commercialize it for that application. Go ahead, Mike.
No, I was just thinking that maybe in a way of producing a more compact, more mobile device and so forth, and kind of shrinking the sizes is one of big kind of tasks ahead of us, of making the labs smaller in size. Mass spectrometry, I worked in mass spectrometry a long time ago, but it is obviously like showed is a big piece of equipment in general. I mean, it's great that they shrank it, but it's technology itself is assuming
it's going smaller with time, but it is still a little big.
All right. Thank you.
No problem. Thank you. I appreciate it.
Thank you so much, Mike. The next is Professor David Crono from University of California, San Francisco. He asked that does the process with using water only work for extracting some molecules that are hydrophobic? David, please open up the mic if you would like to elaborate on your question. David, you are mute. Sorry.
Thank you. I wanted to ask if you can only extract hydrophilic molecules.
Yeah, so that's a good question. A lot of times I will say we detect lipids and people are like, there's no way to detect lipids with water. Well, all of our lipids that we do detect are polar lipids. So they have a polar head group. So they're more like amphiphilic, where they could be extracted in water as well as in organic solvents. If we use an organic solvent, like if we mix it with methanol or ethanol or acetonitrile, it improves the extraction efficiency. And we then start seeing molecules that are truly hydrophobic, like cholesterol or things like that do not like to mix with water. But the lipids and the molecules that we do detect, they are polar molecules. They are soluble in water. They are more soluble in other solvents, but they're still extracted efficiently with water. So there's a mix there. And as I said, as a chemist, we love exploring different solvent systems, but going into DOR, especially the ability to just grab a bottle of sterile water and use it, has really facilitated the translation of the technology. So we decided to go with that for now. Yeah.
Thank you.
Yeah, you're welcome. Thank you for the question.
Oh, sorry, I was mute. So many people actually are very excited when you talk. This is encouraging also for engineers and particularly data scientists to see that how data science can have a real application in clinical field, and you show it. But the question for you is that, do you think that you have amazing numbers, but whether these numbers is still sufficient to apply an AI? And always there is a worrisome that how if that AI overfitting for the samples that you have, and when we are going with more diverse population, different gender, different race, still that may work.
So when people ask me that, I normally say, well, it's so exciting because there's so much work to do. Because it really feels like we could always improve our classifiers. We can always add more samples. And we get asked this a lot, like how do you power your studies? What is the exact number that you need? And I find it really hard, even working with the most incredible statisticians, to get to a number that is like, this is the number and we are locking it down, and it comes down to what the FDA wants to see in terms of a medical device approval. For our research studies, we power them with the information that we have on the delta changes of the molecules and the prediction accuracies that we got in our pilot studies. But as a medical device, we have not started FDA approval yet. But once we do, we've had some preliminary discussions, and the FDA will require, okay, you're going to need like 3 sites, 500 patients each. And then I'm sure when we get there, though, we're going to get into these points, right? We have like different populations, different people that are the devices being used at. So, but yeah, I think at some point, you just make a call. That's what we've done with our classifiers. We thought, okay, we trained it on 200 samples. It's performing at 97% accuracy. We're going to test it out. Let's see how it works. But then the pancreatic example I told you, we then said, okay, now we have operating room data. We're going to add it to our classifiers and they work better. So yeah, I feel like it's like a never ending project, which is good, but it also brings its challenges.
That's amazing. And Scott, actually, he generously offered that if somebody interested to pitch the idea for maybe venture capitalists, he can assist. And maybe Scott, I was wondering that can be part of Instinct, because you're absolutely right. There are many surgeons that they want to see that how they can articulate their idea. And Livia now leading this Instinct, and we have this Shark Tank, and that's definitely we invite everyone who want to be part of it.
Fun.
So Livia, on that aspect, you talked about Instinct. How it's work? Is it just for Baylor College of Medicine? If somebody out of Baylor College of Medicine, engineers, they may have some ideas that can be used on surgical application. How they can be part of that effort?
Yeah, we've been discussing that with the college because of course, the IP issues, right? If we disclose an idea outside of the institution and all of that stuff with invention. So I think that's where it's a little tricky, where we do need to keep some of the confidentiality to protect the inventors and their ideas. But right now we do want to keep it within Baylor College of Medicine, but then there are going to be events that are for the broader community. So we have an event too that I didn't highlight with Rice University, and that's sponsored by Instinct as well. And then we're going to have other events that, you know, I'm hoping to plan with industry, that we would only disclose things that are already being patented and it's safe to do. Some of these brainstorming sessions will be more focused on our own members, so we give them the platform they need to be comfortable to share these ideas.
That's right. Remember at the time of Billy Cohn, actually they created some kind of the NDA that anybody who want to participate on those kind of think tank, they need to sign that NDA. And actually it was very interesting. There was somebody, I'm guessing patent attorney, that was present there as well to give, but building that kind of confidence for the faculties to share, they freely share and exchange the idea is very helpful. And this is what we need, because a person individually cannot build an effective idea. They need to partner with others and partner with industry to make it a scale at large. So I saw that, Livia, you share your email address, please, everyone, if you want to know about this Instinct that has been initiated and led by Livia, as well as her fascinating research, please reach out to her. Otherwise, I'm guessing that if there is no any other question, we can conclude the session today. Again, thank you so much, Livia.
Thank you. I appreciate
fantastic talk. It's super exciting. We should collaborate. I have some ideas.
I know. I still need to come and see your lab, because what we really need are engineers too to help us. So
looking forward to that. Thank you all. Happy Friday, and see you next week. Bye.